Hi, everyone. Hope folks can hear me okay and are doing well in this wintry mix of a day that we're all having. So those of you I don't know, my name is Ellie Van Allen. I'm Chief of Population Sciences. And for the purposes of today, I have the privilege and honor of introducing one of my colleagues and longtime collaborators, Dr. Sasha Gusev. Just a brief primer before I do the formal introduction, just to remind folks to please put Q&A into the box, which we'll be tracking, and we'll save questions for the end of the presentation. Dr. Gusev was a member of the faculty in population sciences, before then received his bachelor's at UConn in computer science, and then both his master's and PhD in computer science and computational biology at Columbia, where he distinguished himself in basically effectively creating the field, both then and subsequently, of transcriptome-wide association methodology and other really just remarkable germline genetics research that have paved the way for just a massive amount of science across human genetics. And lucky for us, has subsequently taken his talents into the world of cancer. Joined the Dana-Farber faculty a few years ago, and has grown a just massively influential and really exciting research endeavor at the interface of germline genetics and cancer across a lot of different domains. And so with that as background and context, we're really thrilled, Sasha, to hear what you have to share with us today about germline genetics and immunotherapy. And I'll let you take it away.
Awesome. Thank you so much, Ellie. I'm very, very honored to be presenting in these seminars, which I've very much enjoyed attending over the years, and appreciate the opportunity. I'm going to be talking about a somewhat, I think, unusual combination of topics, which is my sort of experience and interest in germline genetics, and in particular, the germline genetics of cancer risk and from a common genetics perspective, and then bringing that into our understanding of clinical outcomes, and with a focus on immunotherapy, where I think there's actually a very interesting and natural fusion of those two approaches. So in a little bit more detail, I'm going to start by, because I think this is a somewhat unusual application of fields, I'm going to kind of motivate what we know and what we want to know about the influence of germline variation on clinical outcomes. I'm going to talk a little bit about the methods that we've been applying largely to data that is being collected and curated at Dana-Farber to resolve some of these questions. And I always sort of struggle with whether to talk about the cool results first and then explain the methods, or talk about the methods first. But because this is sort of like a hometown crowd, I think it's actually useful to explain how we do these things. And I'm hoping to motivate folks to apply our methods, take our data, reach out to me about collaborations and in directions that they're interested in as well. And then to the point, I'll present two examples of findings that we've made from analyses of germline data. One where we've identified a specific mechanism of checkpoint inhibitor outcomes, and then another where we've sort of connected components of polygenic risk. So to start with the motivation, what do we know and what do we want to understand about how germline variation influences cancer outcomes? So I think everybody is probably familiar with several decades of work now in understanding the germline influences on cancer risk. Of course, there are classical studies of rare, highly penetrant mutations like the BRCA genes that substantially increase risk, but there's also been an enormous amount of work recently, as you can see in these sort of highlights, of common variation contributing to cancer risk. And in fact, what we're observing from these studies is that for many cancers, the common germline contribution is pretty substantial. And it is also what we tend to call highly polygenic, meaning that there's a very large number, typically of moderate or low individual effect size variants influencing cancer risk. And in fact, to deal with this polygenicity, and I'll get to this a bit later on, we actually started aggregating this variation into polygenic scores to try to boost and combine the signal. So there's quite a bit that's known. In fact, these polygenic scores for cancer risk are now being deployed in clinical trials and investigated in cancer screening for early detection. But from the perspective of tumor evolution and clinical outcomes, I would say that actually there's been, relatively little work in connecting those two pieces together, and that's something that we in my lab are very interested in. But I do want to highlight a couple studies because I think there's already exciting inklings that there is some interaction here. So there was work from Memorial a couple years ago in Nature Genetics doing sort of a broad scan, primarily focusing on rare germline variation and how it is associated with various components of the tumor using, a sort of OncoPanel-like data set that they collect in-house. And I think one of perhaps the biggest findings from that study was that there is an association between carrying germline pathogenic variants and having fewer somatic drivers as a consequence. So something like a two-hit phenomenon where if you already are predisposed to develop cancer, you need fewer somatic pushes to actually transform. There's also, I think, well-established examples of germline variants in BRCA genes, for example, that influence the response to treatment, particularly PARP inhibitors. Again, a great study from Memorial a few years back showing that either germline or somatic variation improved the response to PARP inhibitors in breast sort of related or BRCA related cancers. But I would say that while we now have a pretty comprehensive understanding of rare germline variation, the impact of common germline variation, which is what actually drives the majority of cancer risk, is poorly understood. And most of the reason is that whenever you want to ask anything about common variants, you usually need thousands, if not hundreds of thousands of samples. And so there's been a sort of big data bottleneck in answering these questions. So I'm going to start with talking about how we overcome this bottleneck and then get to how we can then use germline variation to explore interesting biological questions. And the basic idea is that while we do not do sort of large-scale germline genotyping at the moment, what we do a lot of is clinical tumor sequencing. And in fact, I think folks are probably well aware of the profile cohort and project that has been going on for, I think, close to a decade now, doing routine prospective clinical sequencing on a targeted panel, the OncoPanel, for largely identification of drivers and sort of supporting clinical decision making with those drivers. But this has also created, I think, a very exciting, diverse, large-scale research cohort of sequenced tumors. And this figure is a little bit out of date. I looked up in the internal CBio portal this morning, and I think it's close to 40,000 specimens now that have been sequenced and are available as a sort of, I think, an extremely valuable institutional resource for research efforts. But until recently, this was tumor-only sequencing. So the germline variation that I'm interested in was not possible to extract. And even recently, as it's sort of transitioned to a tumor normal project, it is still a targeted panel, and most of what we want to know is about genome-wide germline genetic variation. And so it turns out that we can actually get that kind of data directly from these sequenced tumors. And the way that we do that is with sort of, I guess, a bit of a data trick, which is that when these targeted panels, which include something like 400 or 500 known cancer drivers or suppressors, are sequenced, the intent is to sort of provide high coverage in the targeted exons on the panel, but actually a large fraction, depending on the platform, maybe half of the sequencing material ends up landing in a kind of dust scattered across the rest of the genome. And these are the off-target reads. And this is about in the targeted exons, we have a coverage, meaning a sort of per base pair number of reads on average of over 100. In the off-target regions, we have off-target coverage of about 0.1x, so meaning every 10th nucleotide has one read, so very, very shallow coverage here. But the intuition in this actually came from sort of older efforts to do inexpensive genotyping in the field, is that when you have these one or two shallow coverage reads that you want to use to recover germline variants in the rest of the genome, you can do that by integrating in external reference panels. And that's because individuals share genetic material with recent common ancestors, and they share that material in haplotypes. So there are, for any given target individual, there is some haplotypic sharing to a reference panel of genomes. We use the 1,000 genomes reference panel, but now there's, with TopMed, there are hundreds of thousands of genomes that have been sequenced for these kinds of reference panels. And there are algorithms, and we developed a pipeline to do this in-house that can map these sort of one-off or two-off off-target reads, look at the allele that's captured in this read, map it to the haplotype reference panel, and then based on the sort of co-occurrence of all of these through the haplotype, infer back the likely germline variant that these individuals carry in the sites that were not covered, which is going to be most of the genome, and also actually improve the genotyping of the sites that were covered but had shallow coverage. And so what we get at the end of this process is we've mapped an individual to the reference panel. We get these probabilistic outputs of what their germline genotypes are likely to be. And so this is great in theory. It seems like there's potential here. The obvious question is, how well do you actually recover this kind of germline variation? And so we linked the real OncoPanel sequence data in the profile to germline genotyping that had been done as part of the MGB biobank. Again, one of the sort of advantages of having lots of biobanking and genotyping efforts happening in the Longwood area is that incidentally, there were about 1,000 patients who were participants in both of those studies. And so we were able to use the germline genotyping from blood and compare it to this sort of off-target imputation that we used in the tumors, and evaluate the accuracy of those two techniques. And we see that on average, so each of these dots represents a window of about 100 polymorphisms, common polymorphisms across the genome. On average, we have a correlation of 0.86, I think, or 0.87 between the tumor imputed variant and the ground truth germline genotype. And 0.87 from a genotyping perspective is not great, but from the perspective of basically taking tumor data and getting back out of it genome-wide germline data for free, aside from the compute cost, is pretty fantastic. Essentially, you can think of this as an equivalent study that's 86% smaller, or 14% smaller than if we had genotyped directly. Basically, this correlation translates into the sort of anticipated loss in power. And the other thing to note here is that even though these targets are in specific regions of the genome, there's actually enough off-target coverage that we can impute with pretty high accuracy across the entire genome and get genome-wide, reliable genome-wide estimates. Where there's gaps, it's usually just in the centromeres or the telomeres, which are, in general, difficult to map and tend to have lower coverage. Okay, so that's all kind of a preface for how we get the data just to resolve this problem of how do we actually integrate germline genetics into the clinical questions that we want to ask. And now getting to the actual clinical questions. So there's a couple of ways that we can think conceptually about how germline genetics influences clinical outcomes. One is maybe the most straightforward, is that individuals who carry a certain polymorphism may have different outcomes. So the green allele carriers here have longer survival than the yellow allele carriers, or a shorter or longer time until some event. Another way that this can happen is through the aggregation of risk across many variants into a polygenic score. So individuals who carry lots of a certain directionality of alleles maybe have longer survival than those who do not. And so I'm actually going to talk about both of these because I think they point to interesting ways to study the data. And specifically, I'm going to talk about specific germline variants we've identified that are associated with immunotherapy toxicities. And then I'm also going to talk about some sort of genome-wide causal inference that we've done to connect risk, polygenic risk for thyroid conditions and demonstrated that risk translates into thyroid toxicities in the ICI setting. So a sort of variant specific and a genome-wide analysis. And I hope that I can get through all of those. So starting with the first and I think more intuitive example, can we identify germline polymorphisms that are associated with immunotherapy outcomes? So stepping back a little bit, again, I'm sure, I think this is not news to this audience that immunotherapy and specifically immune checkpoint inhibitors have been remarkably successful in a number of cancer types, in some cases, particularly for sort of advanced tumors leading to cures. And these treatments, these therapies work through a variety of mechanisms, essentially either inhibiting or disabling the inhibition of the immune system or reactivating the immune system through the immune checkpoint process. The other side of this coin of effective therapies, I think we've now appreciated more and more, is the increased rate of adverse events, and specifically immune-related adverse events that these patients develop. And now, as we've had more and more data, this cartoon kind of highlights that there is both a preponderance of acute toxicities. So the majority, almost all patients develop some kind of a toxicity to immunotherapy, depending on the cancer type and the combination of treatments. Something like 20 or 40% develop moderate to high-grade toxicity, and even high-grade toxicities that require that necessitate stopping therapy. Again, depending on which treatment you're talking about, maybe 5 to 10% of patients develop them, and then something like 1% can result in actually fatality. And so this is a common event, and it continues to be a common event as you go up the sort of grade scale. It also, as we're now kind of learning, can become a chronic event for some patients. I think this is still quite mysterious as to why certain individuals develop chronic toxicities, but it does mean that in the process of treating them, we may actually be sort of inducing a lifelong autoimmune condition. And so understanding the mechanisms of these toxicities is very important because on the one hand, we have this really effective treatment. On the other hand, we have these side effects that get in the way of administering that treatment. They make it difficult for us to know when to rechallenge patients, for example. And now, as checkpoint inhibitors are moving into the adjuvant setting, they sort of pose a dilemma as to whether for patients who are in remission, it makes sense to take this risk, roll the dice on some acute or chronic toxicities but within the hopes of getting some of the benefit of the immunotherapy. So I think this is an important question, and it is also one of sort of growing importance as checkpoint inhibitors become more widespread. So there's been a lot of interest in understanding how these toxicities develop, and the little slice of this pie that we're focusing on is host genetics. And the reason we think that this is a practical approach is because a lot of the adverse events present very similarly to known autoimmune conditions, so rashes or diarrhea, IRE, hypothyroidism or colitis. And also we know that those autoimmune conditions in the general population oftentimes have very strong germline components. So we can sort of put the pieces together that perhaps host genetics plays a role in modifying or increasing these phenomena. And we're not the first to put these pieces together. So there are some work again, a few years back, led by Diego Chow and Tim Chan, that explored associations between the human leukocyte antigen, the HLA, which is where most of the sort of heavy-duty autoimmune machinery in the genome is happening, and showing that germline HLA alleles were associated with overall survival, nominally, I should say, associated with overall survival in some of the very early melanoma ICI treated cohorts. So this is very exciting. But this was sort of then extended into thinking about actually HLA homozygosity at the germline level, or evolutionary divergence as they quantify it here, and started kind of opening up a world of possible germline correlates with ICI survival. But as studies have grown and grown, a number of these findings have not replicated or appear to be very fragile to the specific clinical context and or time point that they were measured in. So this is work from Kevin Litchfield a couple years ago in Cell, where they investigated a number, sorry, the slide's a little bit messed up here, but they investigated a number of host factors associated with ICI survival across these cancer types, and none of them were statistically significant except for over here, this is sex. I think women were more likely to develop toxicities. And of course, we don't need generally whole genome sequencing to figure that out. And then more recently, there was a massive study aggregating data from 3,000 or more patients in immunity from Alex Snyder, Matt Hellman, and first author Aparna Chibber that tried to replicate these germline HLA associations, and again saw no effect, and really, I think, quite definitively ruled out the possibility of a pan-cancer effect given the sample size that they had. So there's been kind of a back and forth. The pendulum has been swinging. Initially, it seemed like we'd struck gold in identifying these HLA associations, and more recently, as studies have grown, those associations, either were false positives or were very specific to the time and place in which they were identified. And so our goal was, and this is kind of, I guess, our maybe blunt way of thinking about this, is that we need large scale studies to do these discoveries. We need a lot of data, and we also need rigorous definition of the hypothesis and replication data sets to identify these mechanisms. So we're really keen to set up a large cohort and investigate this question of germline influences genome-wide. And so we did that using the technology, the imputation technology that I described previously. I guess large cohort actually kind of depends on who you ask, but we were able to impute germline information from about 2,700 patients at Dana-Farber who were treated with immune checkpoint inhibitors. And additionally, because this is the profile, we had data from about 24,000 patients who had been treated with other therapies that we could use as a normative group. This is, I think, kind of cool from a statistical perspective, that we can, whenever we find something here, we can also ask if it's just some generic artifact or maybe general phenomenon in the rest of the cohort. We additionally partnered together with folks at MGH, specifically Carrie Reynolds and Eugene Semenov over there, who had very, very carefully collected and genotyped and annotated toxicities from about 375 patients through their unit. And then also we had sort of a really nice collaboration with folks at Roche for targeted replication in their clinical trial data of about 2,500 patients. So we put together a large cohort to do discovery and replication. And just a little bit more detail. So because we were aiming for sample size rather than specificity, we were looking at data from a large number of cancers, essentially anything that was being treated here that had more than 50 individuals per tumor. As it happens, just because of the sort of patterns, the majority of that was PD-1, PD-L1 monotherapy. And then working together with Tony Schwery and Matt Friedman and an incredible team of clinical fellows who helped us annotate the toxicities in these patients through a combination of sort of algorithmic approaches and, of course, manual chart review to identify grade 2 or higher adverse events, as well as grade 3 or higher adverse events in this population. Put all that together, and now we can do a genetic scan for associations. Sorry, this is the details on the on the IRAE definitions, but again, we're mostly talking about medium or high grade events. And so the scan looks like this. Every dot here is a polymorphism that we tested for association with time until you develop a toxicity. We model this in a survival model with the competing event, or really a multi-state process, so that patients who live longer are not do not induce false positives in the associations just because they have more time to develop toxicities. So this sort of properly manages the potential survival bias here. And for each of these points, we test, and we have an established threshold of genome-wide significance. And what we saw in this cohort was 3 independent loci that were significantly associated with toxicities. With the one that I'm going to focus on the most is this association near the IL-7 gene, which we were then able to replicate and sort of functionally characterize, I think, in the most meaningful way. But just at a glance, we already see some striking evidence of germline variation influencing IRAEs. And I should say that these effects were seen for the medium to high grade events. And I think mostly it's a power issue where the number of very high grade events we had was relatively small. So we couldn't rule out whether the association was there or not. I think a question you could be asking is, how specific is this? You had this cohort that was a kind of all comers pan-cancer analysis. Well, to our surprise, actually, when you look at the association near the IL-7 locus, for example, it is nominally significant in 6 of the cancer types that we investigated. And in fact, essentially in all of them, they are not, significantly different from the mean effect, which is a hazard ratio of 2, meaning per instance, individuals have about a 2-fold risk of developing a toxicity if they carry this risk allele. And so this appears to be a genuine pan-cancer phenomenon, although there are interesting patterns here. And, every time I present, we come up with some possible stories for what these patterns mean, why kidney, bladder, and melanoma tend to have stronger effects than, for example, lung, head and neck cancers. I think, what we're doing next is trying to boost the sample size here to get a better understanding of the effects in individual cancers. But at least a priori, we see that this is quite widespread. And the phenomenon is the same for the second association that I'm showing you here. So as I mentioned, a big component of this analysis was to put together data so that we could immediately replicate the associations. And we see the polymorphism that was associated in the profile for near IL-7 was highly significantly associated in the MGH data. And this is actually cleaner data, and the fact that the signal, the hazard ratio here was stronger, kind of reassured us that we were on the right track. And then working with the folks at Roche, there was a nominal association of the polymorphism in their data. And I don't think I'm mentioning it here, but we've since also engaged some colleagues in the UK in a collaboration. They were able to replicate this variant for association in melanoma. And we've also been working with folks at BMS on clinical trial data in kidney and bladder, and continue to see the association there. So one of the nice things about a germline mechanism is that you have the RSID, the SNP ID for this variant, and you can just ask people to look at it in their data and see if it replicates. It's a very clear analysis. And so far, we see that this is the case. And most of the time, to give you a sense of the numbers here, so this polymorphism is at about, it has a carrier rate of about 7% in the population, so sort of moderately common. And individuals who carry the variant pan-cancer, they tend to, roughly 30 or 50% of them go on to develop toxicities, whereas individuals who don't carry the polymorphism, it's more like 6 or 7% of them develop toxicities. So there's a pretty substantial risk increasing effect just of this variant alone. Now, with these really large effects across multiple cohorts, you could be wondering, well, maybe this is just a known autoimmune phenomena that has nothing to do with checkpoint inhibitors. And so again, I mentioned we have this normative population in the profile that just received conventional non-immunotherapy treatments. Obviously, they're going to be different in terms of their staging and the advancement of the disease. But when we looked for various IRAE-like diagnoses in these patients, we saw no association whatsoever. And again, given the sample size here, we're really well powered to see something. And this has continued to hold as we've gone into the clinical trial cohorts where we have control arms. We see no effect of the association outside of the immunotherapy setting. And IL-7, the gene specifically, while it is well established, an important cytokine and has been connected to autoimmune diseases, this polymorphism, to our knowledge, isn't associated with any known autoimmune condition. So it appears that this effect is really specific to the checkpoint inhibitor setting. And that may explain like why the effect is quite large. For a typical common polymorphism, you don't really see odds ratios of 2 or 3 like we do here. So this has all been thinking about it from the perspective of the polymorphism, but I guess the logical question is, I keep calling it the IL-7 locus, but we don't really know that it is functioning, is operating through the expression or is mediated by IL-7. And again, one of the nice things that we can do with germline data is that we can go to other cohorts that have germline genotyping and gene expression measurements and then ask if this polymorphism is a QTL for expression in those cohorts. And so we use gene expression data from the GTEx. And what we see plotted here is the lead polymorphism from our study, the canonical transcripts for IL-7 plotted in these black lines. And what you can see is that the non-carriers of the polymorphism have sort of, average expression of IL-7, nothing going on here. And then the carriers of that polymorphism see substantially increased expression in this little sort of cryptic position over here right next to the SNP. And additionally, increased splicing. This is through junction reads into these other canonical exons of IL-7. So this is a novel we call it a novel cryptic exon. Was not previously annotated for IL-7, and it is almost perfectly correlated with the polymorphism. You can see non-carriers basically have no expression or very, very low expression. Carriers have high, and the homozygotes actually kind of hard to see here, but are really overexpressing this exon. And so this is not, case closed. There may be other mechanisms going on, but I think this is about as strong associative evidence as you can get for this effect, at least in some sense involving the expression of IL-7, possibly a novel isoform, although we don't unfortunately have long read data here to really interrogate the transcript or the isoform that this exon develops. We've tried all sorts of sort of in silico approaches to understand what the exact function is. I don't think it's not really our area of expertise, and we haven't been very successful. But we see this very, very strong association with the expression of this exon. So that gives us a putative causal gene in IL-7. I think stepping back a little bit, I mentioned that IL-7 is known to be an important cytokine, and this led us down a direction of trying to understand a bit more how mechanistically this increase in toxicities could be happening. So there's been quite a bit of literature. You can see these screenshots are from a while ago of IL-7 being involved in homeostasis of CD8 T cells and in lymphocytes in general. And this we started to wonder whether we could see the effects of this polymorphism more broadly in the patients. And so specifically, we looked at lymphocyte counts, which we can just get out of the CBCs, out of the clinical labs for these patients. And we kind of came up with a lymphocyte stability metric, which is basically just the change in lymphocyte count measured right after the start of immunotherapy versus right before. You see in the carriers, there's no change in stability, whereas actually in the non-carriers, we see a significant drop in lymphocytes after some time after they've initiated immunotherapy. And this interaction between the two is significant. And we also saw, so kind of flip things around here, we saw this both in the MGH and the Dana-Farber cohort. So this story of IL-7 being involved in lymphocyte stability, this polymorphism altering the effects, the expression of IL-7 likely through this novel exon, and then that also being reflected even at the very crude level of lymphocyte counts in these patients, I think all sort of fit together with the biological mechanisms we think should be involved. The other interesting thing, and this is a bit of a, it became a bit of a side story, is that actually lymphocyte stability, this measure that we defined as the delta, is itself associated, even if you just set aside the SNP, is associated with developing slightly more IRAEs. Obviously, the SNP association is stronger, but it suggests that maybe there's actually a broader mechanism at play here beyond just what IL-7 is doing. It is also associated with improved survival for patients who do not develop toxicities. And this is, in fact, in contrast to the polymorphism, which I'll show is to date not associated in any way with progression or survival. So there's some complex dynamics here happening. Lymphocyte stability is playing its role in toxicities and survival. The polymorphism is playing its own role in IL-7 expression and lymphocyte stability. And these are sort of questions that we're, trying to disentangle further. So this brings me to the point that I alluded to. What about the effect of this polymorphism in response? So in the cohorts that we looked at, so in the profile, we have overall survival through the National Death Registry. We see no association, likewise in the MGH data. And then in the Roche cohort and with our collaborators in the UK who have progression-free survival, we again see no association of this polymorphism. So I think we're pretty confident that in the ICI setting, this SNP just gives you toxicities. It's just bad news and does not give you a survival or response benefit. But what's interesting is as we kind of dug around, we went back to some public data, and specifically because we had replicated the association in melanoma in the UK, we looked at public data from the TCGA cutaneous melanoma cohort, which had been collected a long time ago. And actually, a lot of those patients were not on checkpoint inhibitors, even though that's now the standard of care. And we saw in this population, pre-immunotherapy, largely, this IL-7 variant was associated with improved survival and actually with a pretty substantial protective effect, a hazard ratio of about 0.5, highly significant and associated both with OS, PFS, and disease-specific survival, because all that data is available in TCGA. So again, the story gets a bit more complex. It looks like in the case of melanoma, in the checkpoint inhibitor setting, this variant is all bad news. It's just toxicities. But prior to that, in the conventional chemo setting, at least in this data, we see actually it has a significant association with improved survival. And this starts to, and we now, I think, are fingers crossed, may have a funded effort to pursue this in the adjuvant setting for melanoma to try to investigate these dual effects. It may be the case that for carriers of this polymorphism, the survival benefit is very meaningful, and the increased toxicities on ICIs would not be worth it. So we're kind of following this up further. I guess this is all just to say that you can pull on one thread on one polymorphism and you can follow it down a lot of interesting directions, both in its effect on toxicities, but then eventually that brings you back to in certain settings association with survival and implicates novel biomarkers. So this sort of wraps up the single variant approach example that I was talking about. And again, I think I'm not going to run through these bullet points, but I just want to highlight the fact that we can use imputation from tumors in routinely collected clinical data to run these genome-wide scans. And when we do so in a treatment setting, we identify interesting associations. In this case, the association is replicated. And they pointed us to a novel exon of a gene that had not really been explored very much in this setting. They told us something about the potential cell types that are involved and the stability of those cell types. And in a sort of specific way, even implicated some associations with survival. So I think this is a very fruitful way to try to understand kind of causal mechanisms and modifiers of outcomes. So that was all kind of a story about a single polymorphism. And now just in the last couple of minutes, I wanted to touch on this other way of thinking about these associations, which is from the perspective of polygenic effects when we aggregate many variants together. And again, as I noted, for most common traits, polygenic effects are really where it's at. Most common diseases are driven by thousands, maybe tens of thousands of causal variants. And so if we want to bring in an understanding of other phenotypes and how they influence clinical outcomes, we ideally want to work at the polygenic level. And so we can do that by employing what are known as polygenic risk scores. And these are very, very simple. Essentially, if we we run somewhere, typically in a large external biobank, an association study for a given phenotype, and identify effects of all of the genome-wide variants on that phenotype, we can then put together a weighted sum in a new set of individuals and predict the phenotype using all of these weights. So instead of talking about one polymorphism, we're going to aggregate across thousands of polymorphisms, add up all of their little weights together, and get a score for an individual. And so just on a technical level, again, we can do this with the imputed data that we've pulled out of the profile with high accuracy. So this is not the predictive value of the score. This is just how consistent is the score when we imputed from germline SNPs versus when we imputed from tumors. It's actually very consistent. The consistency improves when we go to the polygenic level, and it's just slightly deflated. There's sort of no outliers here, so we can kind of proceed fairly directly with these associations. We do also confirm that the known polygenic scores for cancer risk are indeed elevated in those populations in the profile. So profile patients with breast tumors that we imputed have a substantially higher germline PRS, polygenic risk score, for breast cancer, and so on. And actually, in some of these cases, the association is quite strong. So this is interesting in and of itself, and we're kind of pursuing ways in which cancer risk may be altering effects in these patients. But here it's just a proof of concept that we do see an association with the label that we know. And now we can turn to new phenotypes. And again, as a proof of concept, the phenotypes that we turned to were putatively linked to toxicities for checkpoint inhibitors. So specifically, we were focusing on associations with, or the genetic sort of predisposition to thyroid conditions or hypothyroidism. So there's been a number of genome-wide association studies in a normal population, in particular in the UK Biobank, a population of about 500,000 individuals in the UK that were just kind of routinely collected and genotyped. And these association studies have, as I mentioned, identified a very large number of genes associated with hypothyroidism in this UK population. Additionally, these genes can be put together into a polygenic score, and that score is fairly predictive of hypothyroidism in held out data. So you can see here like deciles of the score and the odds ratios of developing hypothyroidism depending on your decile. So when we aggregate all these effects, we get a pretty meaningful predictor. So we took this idea and we moved it into the ICI setting, and we worked together with colleagues at UCSF, at MSK, and at Vanderbilt to put together 3 cohorts of non-small cell lung cancer patients on immune checkpoint inhibitors. Again, this data is the imputed data that I talked about from the profile, which at this point in time included about 600 patients. And we asked if this polygenic score was associated with the development of thyroid IRAEs, which are very easy to define just based on clinical labs, mostly easy to define. And in fact, we see in both cohorts a significant association between the PRS, so higher um, uh, uh, germline predisposition to hypothyroidism means faster development of thyroid IRAEs on these checkpoint inhibitors, consistent effect in our cohort as in their combined cohort. And actually, as it happened, I guess lucky for everybody, there was a concurrent study going on at Genentech with their data, and they also saw the same association in a completely independent analysis. So a hypothyroidism polygenic risk score, they've just flipped around the axes here, but is associated with more thyroid IRAEs than the low polygenic risk score. So this is all just to kind of put, I think, on folks' radars that we can do scans to identify novel associations, but we can also take pre-existing hypotheses, like maybe your predisposition to hypothyroidism has some influence on what happens to you once you receive treatment, and then identify these associations through those hypotheses. And so with that, I'll close with the overall conclusion. So, I think mostly I just want to get folks excited about the idea of extracting germline data from the clinical cohorts that we work with. This, while we're doing it here, we've started working with the team at AACR, Project Genie, which is doing this at other institutions. This can be done with data from Foundation Medicine or from Tempus. Essentially, any targeted panel can be imputed in this way. And then now you have basically a germline cohort built into whatever study you're conducting, and you can look for these sort of somatic and clinical associations. We have been running scans to identify associations for immunotherapy toxicities, and we've identified some which I think have implicated interesting biology. We've also been doing this, as I mentioned, at the polygenic level. And this is just a start because there are, I think, both interesting causal questions that we potentially are now able to answer. Again, if you have a hypothesis that the predisposition of a certain condition may be implicated in a certain clinical outcome, we can test that now with the genetic score. Additionally, if you want to know if toxicities, for example, are associated with some downstream effect, we now have a kind of causal lever to probe that question. So we have a polymorphism that increases your rate of adverse events, and now we can ask what else does that increase in these patients. So this starts to get at questions related to causal inference in the clinical setting. Again, just to kind of tease the idea that there's a lot of other events outside of immunotherapy, pan-cancer and within cancer that have a putative germline cause that we may be interested in exploring in the same way. And I'm always happy to chat with folks about that as well. So with that, I just want to acknowledge the many people involved in leading the work, in particular, Stefan Goha, fantastic postdoc and Inte Moon grad student who led these studies, as well as the team working together with Tony Schwery, Matt Friedman, Ken Keel, our friends in the UK, Ben Fairfax, Deb when she was here, and of course, Ellie. And with that, I'll take your questions.
Sasha's really brilliant talk. It's like fascinating science. I had queued up a bunch of questions in my brain to ask you, but luckily for everyone, probably, we're actually getting some great questions in the chat, so I'll just jump to those. So the first question is, there's a two-parter. Do you expect we could find somatic mutations that influence response to treatment or outcomes? And then two, is there a trend to move to whole transcriptome exome sequencing instead of targeted panels, perhaps for such kinds of questions?
Yeah, that's a great point. So we did, as it happens, also run a scan of all of the somatic data here for whether it was associated with toxicities. And in this pretty large cohort, we didn't see anything at all. And in fact, it's like a supplementary table buried somewhere in the paper, but in principle, it was quite a lot of work to try to explore that angle. So I don't know, I'm biased, but I suspect that in the case of toxicities, host genetics seems to be playing a more important role than somatic variation. I think, and we can kind of continue to probe different ways that somatic variants could be associated with survival and progression. It's hard. And I think in part the patient heterogeneity and sort of getting a handle on the stage, the disease progression, and the disease burden that these patients has makes it difficult. So I wouldn't say we have anything conclusive there on the somatic side. We do have instances where germline variation seems to alter the somatic landscape. And I think probably the most concrete example of this is that individuals who we find to be at high germline risk for breast cancer tend to have significantly fewer TP53 mutations and a generally sort of better responding and less aggressive tumor. And so this interplay between if you have a lot of germline predisposition, maybe you need less somatic sort of nastiness seems to be a recurring theme, but we're in really early stages there. To the second point about other kinds of omics, I mean, I would love to bring that in. That's, as Ellie mentioned, my sort of background is in transcriptomic studies. And, I think thinking about the perspective of ideally what we would have is the expression or even the proteomics of IL-7 here, and we can ask how those polymorphisms affect this sort of whole transcriptional cascade. But we primarily just work with the clinical data that we have. So most of the time when we've brought in other data, it's publicly available bulk or single cell sequencing where we can then try to find this SNP and ask a specific hypothesis. I guess one thing I'll mention is that this imputation approach does work on RNA-seq as well. And so if you have a transcriptomic data set with no germline, we can get the germline back out of it. And that maybe opens up some opportunities.
So I suppose a semi-related question around data is, so there's a question, how are the germline TCGA melanoma data obtained? Did you have to recall all tumor-matched normal pairs in genomes or exomes?
Yeah, so it was a bit of a headache, and I'm happy also to share that data with people. We recalled and re-imputed the germline genotypes off of the SNP arrays that they had done for primarily CNV detection. So they had these like AFI 500K arrays from when I was in grad school, and they had analyzed them. I don't know if people remember, like Bird Suite was the tool that I think Steve McCarroll developed. Anyways, there's all these flashbacks going through that data, but yeah, we basically had to go back to their CNV panel, which they had done in tumor normals, and then recalled it there. Now we have nice clean VCFs, and if somebody needs them, we're happy to figure that out.
Wonderful. All right. Oh, question from Miles Brown. Have you explored other genes involved in ICB toxicity?
Yeah, so that's a great question. We have those two additional associations, which we have not probed much further. I should say that as we've brought in more data since that original study, and we see that the effect of IL-7 in the the newer profile patients is even stronger, but the effects of those other associations seem to be moderating. So we're not quite sure what's going on. We are now, I think we're hitting the limits of what we can find in one institution, and we're working with other folks to do sort of, a pan cross-institutional consortia type effort. But that, of course, takes like 5 times longer. So we're still waiting on those results.
Cool. All right. So next question. Sorry, Sasha, I'm just going to run them down. This rapid fire.
Yeah.
Have you investigated whether presence of germline mutations, maybe I think is associated with lower somatic driver mutations?
Yeah. So I guess there are those findings at Memorial, and those are specific to rare pathogenic variants. So that I think is fairly well established. Because we're working with common variants, we don't see as much of that, except for this one example that I mentioned where common breast cancer risk is associated with fewer somatic drivers. So I would say that this is not, there are instances where this happens. It's not a pervasive phenomena, this two-hit effect. I think in the literature, it's also been observed for prostate cancer as well, where higher risk tends to correspond to, I mean, there's not a lot of somatic SNV drivers there, but tends to correspond to less aggressive tumors. So maybe the hormonally driven cancers, this is more common. When we did a big scan across all of the cancer types, we didn't see this as a pervasive thing.
Wonderful. Here's Sasha. This one you'll enjoy. Are germline SNPs or PRSs, polygenic risk scores, currently being used for clinical decision making in any setting?
That's a great question. So there are almost all of the work has been done on the screening side, on the risk side. There are tools that the public can use to evaluate their cancer risk, and primarily this has been done. There's a couple of platforms. I think Predict is what they're called in the UK, where you basically can upload your PRS and get a fairly rigorously validated prediction of risk in addition with a bunch of other features. So that's ongoing. Obviously, there's 23andMe and there have been other commercial vendors kind of trying to start rolling this out. I think it's at a bit of a standstill. There is an effort by the NIH that's called the eMERGE network, which is running clinical trials of specifically this question: give people PRSs, look at their risk, follow them up. And I think once those trials start to get read out, this will be incorporated. I will say we're very excited about the possibility of using these, especially the IL-7 association, which again is like one of the strongest hits we've ever seen in a study, and thinking about how it could be integrated, particularly in the adjuvant setting, but that's all still very aspirational.
Cool. There's a few questions that are a bit technical. I just because there's actually a really, I'm going to skip around a little bit. I apologize for folks, but hopefully we'll have time. Does it make sense to calculate PRS using SNPs identified in different racial cohorts?
Yeah, super important question. There is a well-established bias in these PRSs as a function of the population they're collected in, and we restricted all of these analyses to European ancestry individuals. It's kind of unfortunate. We basically have to do that to make sure that we don't introduce biases. That's changing, but certainly ancestry is a component. And I didn't talk about it at all, but we have inferred genetic ancestry in these cohorts, and we sort of have other efforts related to that.
So, and if a patient uses a commercial genotyping service, 23andMe, whatever, can that data be used for your analyses?
Yeah, absolutely. I mean, that's even better. We're trying to get that data through the back door, but that it's trivial if they have a commercial genotyping like 23andMe or all of us or any of these other efforts.
Let's see. I'm trying to think through the. So Sasha, do you want to eyeball? There's a technical question. I don't want to read the whole thing.
Oh yeah, let me see.
Is it use of off-target reads and imputation algorithm with low-pass sequencing? I mean, you want to have it?
Yeah, so I think, we have a paper on this, which I recommend. So it's in Genome Medicine. I'm actually the first author. It was like the last first author paper that I wrote on the imputation. In the OncoPanel, about half of the reads are off target. But that doesn't tell you, I would say that 0.1x and even as far as low as 0.05x coverage has been good enough for imputation. In general, we've thrown this approach at multiple different sequencing platforms, and we're always able to impute with decent quality. So I've yet to see an instance, if you have more than a few hundred genes, that you can't do good imputation in this approach. If you know the off-target coverage and the uniformity of that coverage, that's going to drive the whole the power.
And then, last but not least, do you think the type of immunotherapy influences results? So PD-1, PD-L1, PD-L2,
Yeah.
or CTLA-4? or
Yeah, I mean, you
other?
saw that table. I think it's a fantastic question from Miles. The table where we like 90% of our cohort is PD-1, PD-L1 monotherapy, so we couldn't really explore it. In the UK data, we saw the effect start to wash out with combination immunotherapy. And so I do wonder if at some point the treatment dose is like so substantial that the germline genetics actually starts to matter less. But that is just a hypothesis that we've seen in one cohort.
Wonderful. So there's some really fantastic questions, a brilliant talk, great science. I think we're ending right at the hour. I think as directed. Thank you all for attending. A great participation. And Sasha, wonderful science as always.
Awesome. Thank you so much. And reach out. Feel free to email me if you have any other thoughts.
Great.
Great. Thanks, folks.